Greetings, ladies and gentlemen. I'm Rahagir from University, Bangladesh, and today I'll be presenting my work on the sexual differences in fat cell gene expression profiles when subjected to dietary restrictions. So, caloric restriction and dietary regulation have been integral aspects in aging and metabolic research for decades. Caloric restriction is well proven to exert beneficial effects in health and longevity in aging individuals through multiple nutrient sensing energy homeostatic pathways, particularly the AMPK-mTOR junction, as well as epigenetic regulators like the serotonin-FOXO networks. However, the degree of these benefits is substantially skewed with respect to sex. Generally, females are more resilient to environmental stress and have stronger longevity metrics when subjected to anti-aging approaches. However, males have been reported to gain more anti-aging benefits from caloric restriction in some recent contradicting studies with aging biomarkers. One of these major biomarkers is adipocytes or fat cell accumulation. Adipocytes provide thermoregulation and endocrine balance in the body, but buildup of unnecessary adipose tissues can hinder health and longevity via caloric dysregulation, loss of homeostasis, and ultimately acceleration of aging processes. Again, there is substantial divergence with sex in the metabolism and regulation of these fat cells with various forms of fasting and dietary restrictions, but a gap of understanding remains in the fundamental genetic and molecular mechanisms involved in caloric restrictions, effects, and their sexual biases in the adipocyte metabolome. Which leads us to a primary hypothesis that the expression of a set of sexually divergent genes can be associated with this phenotypic dimorphism, and secondly, by extension, these genes modulate different pathways in males and females in the adipose tissue transcriptome, leading to these phenotypic differences. Here is a brief outline of our experimental procedure, acquiring public data from microarray sets, processing them, and analyzing them for sexually divergent DEGs. Moving on to our findings, analyzing 6 different publicly available microarray datasets, we identified 34 most significant differentially expressed genes or DEGs in the aged adipose tissue with a substantial sexual bias, 7 of which were biased towards females and the rest were upregulated for males. Here in this volcano plot, we see the folding change, which is greater than 1.0, and significance levels of less than 0.05 of the sexually divergent DEGs, with X-inactivating transcript being the most significant for females and Y-linked taxolin gamma pseudogene for males. With these identified DEGs, we went on to perform KEGG pathway simulations, which revealed male-specific upregulated genes to be significantly associated with demethylation, dealkylation, and post-translational protein modifications. Contrastingly, female upregulated DEGs are affiliated with fat metabolism, cellular growth inhibition, and intracellular signaling. Another major aspect of male upregulated genes lies with epigenetic regulation of chromatin proteins and histone modification sites, as visible in this gene ontology network, with nodes representing function and edges showing number of overlapping DEGs. Notably, aging and rejuvenation are associated with molecular mechanisms which substantially depend on demethylation of histone proteins by sirtuins and FOXOs, and the accessibility of gene expression sites contribute to the overall cellular homeostasis in the adipocytes. Finally, the enriched pathway studies are further validated when we constructed a protein-protein interaction map with the DEG products. 3 major clusters of protein interactions were elucidated from the DEGs. The ATP-dependent RNA helicase associated cluster shows dimorphism in splicism formation and protein modifications as a response to caloric restriction, while histone demethylase UTY establishes a second cluster with FOXOs and sirtuins in males. Finally, a large intercommunicating cluster is observed with X-inactivating transcripts in females, with that of CD signal transducers and JAK-STAT-associated apoptotic network. And finally, from these data, we can infer that caloric restriction does provide benefits to both males and females, but the underlying mechanisms vary greatly according to sex, with aged females benefiting from fasting with regards to fat metabolism and adipose tissue recycling, while males can attain cellular homeostasis in the adipocyte tissue microenvironment through caloric restriction, but have relatively low effects on fat metabolism. And here are some excellent references that we recommend on caloric restriction, adipocyte regulation and metabolism, as well as sexual dimorphisms in aging. And with that, thank you for your kind attention and patience. Please stay safe, stay healthy, and take care.